Okay, I'm gonna really quickly, I'm gonna hopscotch around this PowerPoint. So usually when we get to the end of water properties, I do a little recap of all the properties. Remember that for the properties of water, you need to know the name of the property, right? The term, the definition, the biological significance or the application of it, and then be familiar with the examples that we talked about. Right? So you need to be familiar with 4 things for each of the properties of water. Normally, I would do a little recap once we work through them, but I'm going to do the recap to begin with as a review, right, of what we talked about last class, and then pop back and finish the last property that we didn't get to talk about on Thursday, the solvent properties. So we talked about cohesion, right? That cohesion property of water is that tendency of water to form hydrogen bonds with other water molecules versus adhesion, which was the property that was forming hydrogen bonds with other ions or polar molecules. Remember that cohesion itself results in surface tension, and we talked about the importance of surface tension in biology. And then we can also see that cohesion and adhesion will work together for capillary action. And that's, remember, when water can now move through small spaces, wicking through, shimmying through the small spaces without having to use any ATP energy or any cellular energy. And we talked about how that's important for plants because then they can transport water without having to spend energy, right? Allows them to reduce the cost of surviving. Then we talked about heat and atomic motion, right, and how that's associated with heat. And then we talked about how high specific heat and high heat of vaporization are 2 different ways that we can use water's properties to regulate temperature. So we talked about high specific heat, which means that because water has all those hydrogen bonds formed, it resists moving, right? So in other words, it resists warming. So it helps to moderate temperature in the environment, right, where there's water around, and it helps to moderate temperature in our bodies, right? Conversely, if you have an arid area like a desert with very little water, the temperature is going to fluctuate quite a bit, right? So high specific heat, when there's water around, you moderate temperature. We talked about how it's cooler at Santa Cruz than it is at West Valley, and that's because there's a big body of water there absorbing the solar energy and moderating the temperature. We said we can take all those hydrogen bonds and break them using energy, right, as a mechanism to get rid of extra heat energy, right? So we can vaporize water, breaking all of its hydrogen bonds to turn it into a gas as a way of eliminating excess heat. So high heat of vaporization, we talked about how we use that in evaporative cooling, right? Which is sweating is a type of evaporative cooling. Keep in mind that sweating's not the only form of evaporative cooling. Panting, gaping, and birds are also ways that you can evaporatively cool. There's even crazy modes of evaporative cooling like urohydrosis, right, which is what turkey vultures do, where they poop on their feet, right, and then the liquid component of the uric acid that they're secreting evaporates away. It's kind of their equivalent of sweating by pooping on their feet. Crazy. Urohydrosis. Anyhow, density properties, we talked a little bit about how water gets more dense as it cools until it hits 4 degrees Celsius, and then it forms maximal hydrogen bonds and turns into a solid ice, right, which gets less dense. And we said that when water goes from a liquid to a solid, it gets less dense, which is very unique because most things, when they go from a liquid form to a solid form, get more dense. Water's the opposite of virtually everything else on Earth. Right? Okay, so that's where we left off. Does that sound familiar? Any questions on any of those properties of water? No. Okay, so I'm going to finish up with talking about water's solvent properties. And we talked a little bit about this already when we were learning the terms solution, solvent, and solute. Remember, we said water is the universal solvent, which means that it's a good dissolver. It's a universal solvent because, again, 70% of Earth's surface is water, 70% of your body is water. So chemical reactions happen in water, right? So water is a really great solvent. The other thing that makes water a really good solvent is the fact that it's polar. It's going to interact with other molecules that have polarity or charge on their surfaces, or are ions. And as water interacts with those charged molecules, it's going to help to dissolve them. So if we imagine sodium chloride, right? So here's a little representation of a crystal of salt made up of sodium and chloride ions. What's going to happen when you put that in water, right? Remember that sodium's got a positive charge and chlorine has a negative charge. The water molecules that are in that glass, right, are going to start to interact and be attracted to those ions. And if you take a look, right, sodium, what's going to happen to it is water's going to come in and bust the bond between sodium and chloride. And notice how the slight negative charge end of water molecules are going to be attracted to that positive charge. And they're going to come in and start interacting with that sodium all around it, right? Surrounding it. And in that process, suspending it or dissolving it, in other words, right? And the same thing's happening with the anion, the chlorine. The negatively charged ion, the slight positive ends of water molecules are going to be attracted to that negative charge, and they're going to start to interact with it and surround it, essentially isolating it and dissolving it, right? So when something's dissolving, it's interacting with water in this way, right? So you can, does that make sense? Suspending it, dissolving? Right. Okay, so you can define molecules or their interactions with water using two terms, right? So anything that's polar or charged, it's going to interact well and get dissolved well in water. We call it hydrophilic, right? So in this case, hydro means water, philic means loving, right? So hydrophilic is going to be any molecule that has a charge, that is slightly polar, that is an ion, is going to be hydrophilic. They're going to interact well with water, right? Those slightly polar ends of water. So hydrophilic means water loving. Oh, we love interacting with water. Conversely, anything that's nonpolar, in other words, overall neutral charge, not charged at all, is not going to like to interact well with water, right? Doesn't have a charge, and so doesn't like that, slightly polar nature of water. These things are what we call hydrophobic, water fearing phobic, right? They don't interact well with water. So let me ask you this. What do you think about sugar? Is it hydrophilic or hydrophobic? What do you guys think about sugar?
Hydrophilic.
Hydrophilic. It dissolves well in water, right? What do you guys think about cotton?
Hydrophobic.
Hydrophobic. It doesn't mesh well with water.
Cotton? So how many of you guys have had a nice, lush, high-end cotton towel versus the cheapy Motel 6 towel? You ever notice the difference in absorbency? Some towels will dry you off really nicely and other towels will not. So cotton made out of plant cells, hydrophilic. Plant cell walls are hydrophilic. So like paper towels made out of plant cell walls are hydrophilic. Cotton towels, hydrophilic. It's when they have polyester blended in there a lot that they don't absorb well, because polyester, guess what?
So materials that absorbs water will be hydrophilic.
Mm-hmm.
Got it.
Yeah. They interact well with water. How about oil?
Hydrophobic.
Hydrophobic. Yeah. Indeed, right. does not interact well, will clump away. And we'll talk about this concept a lot more when we start talking about what's going on inside of our body, right? So things that dissolve well in water will be readily transported in your blood vessels, right? Because remember, blood is mostly water. So what happens, do you think, when you have a big fatty hamburger with French fries and a milkshake? Happens to all those lipids you eat in your blood vessels? Hydrophobic lipids, right, in a watery blood solution can tend to be repelled and glom, try and get away from that water, building up in your blood vessel walls instead. But we'll talk about that later. I'll gross you out with cardiovascular system pathology later. Any questions on water and its properties?
Sorry, I just want to clarify, for example, the hydrophilic is the cotton, and then the hydrophobic is the polyester?
Yeah.
Okay.
Yeah. So Gatorade powder, what do you guys think? Hydrophilic or hydrophobic?
Philic.
Hydrophilic. How about chocolate milk powder?
Philic.
Hydrophilic. Interacts well with water. If it dissolves well in water, it's hydrophilic. If it doesn't, hydrophobic. Gore-Tex on your fancy outdoor jacket.
Hydrophobic.
Hydrophobic. Yeah. All right. So sounds like you guys are getting it. We're going to move on and talk about more complex molecules. So water is considered to be inorganic at the chemical level, and that's because it doesn't contain carbon in it. It's just oxygen and hydrogen. What we're going to do for the rest of our molecule lectures, talk about the organic molecules, the molecules that have carbon in them, right? So organic in this context means has carbon as part of the molecule. And we're going to talk about much larger molecules now. The biological molecules are going to be significantly larger than water molecules. Water molecules, just 3 little elements. We're going to start talking about the macromolecules, the big molecules that are in our body that are made up of hundreds, if not thousands, of chemical elements, of atoms, right? So they're much larger. And I'm going to walk you through an example of how we build macromolecules. We're going to talk about what are called monomers versus polymers. A monomer is going to be like a building block for a macromolecule, right? It's going to be a small aggregation or a smaller aggregation of atoms, right? It's like, I'm missing the lecture hall because sometimes I use a lecture hall as my example. So in the lecture hall, I don't know if you've been in the bio lecture hall, but it's got this great big brick wall, right? And if I want to build a great big molecule or a great big wall, I can use building blocks to speed up my process of building that wall. I can go to the hardware store and get pre-made bricks, right? Stack them up and build that wall pretty fast. That makes sense? A brick is like a monomer, right? And that's faster than if I had to go and make all the bricks myself. If I had to harvest the sand and grind the rocks to make it, if I had to harvest the pigment and then figure out some type of mortar to get the bricks to stick together and dry them, right? So by buying the pre-made or using the pre-made bricks, I speed up my process of making that big structure. Monomers are the same principle, right? You're going to be using monomers, molecular building blocks, molecular bricks to build larger molecules. So that's going to allow you to streamline the process of building these large macromolecules that are important for our bodies, right? So does this make sense? And when you put a bunch of monomers together, you get a polymer. So a polymer is 2 or more monomers together. Right? It could be 3 monomers, or it could be 1,000 or 100,000 monomers. You're looking at a polymer. Yeah. Make sense? Monomers versus polymers. Monomers are molecular building blocks. They're bricks. They are the subunits of our big macromolecules. And so what we're going to do is we're going to talk a little bit about how we use monomers. We're going to learn our categories of organic molecules. And for each one, we're going to talk about what is the component monomer. So for example, for building a protein, the monomer is going to be an amino acid. For building a carbohydrate, the monomer, the building block, is a simple sugar, right? A monosaccharide. So for each of our molecular categories, we're going to talk about what is the characteristic monomer. You're going to be familiar with that. But I'm getting ahead of myself. Let's first talk about these two chemical reactions, right? And these are the ones I want you muttering in your sleep by Sunday. Dehydration, synthesis, and hydrolysis. And again, we're going to be talking about these two types of chemical reactions that happen in biological systems for the rest of the semester, right? So the sooner that you get a handle on them, the easier the rest of the material will come for you, right? So we're going to use our word roots to help us break down these terms to make them a little bit more intuitive so that you can work it out when you see the term. I call your word clues, right? What are you doing when you're synthesizing something? What are you doing when you're synthesizing something?
You're making it.
You're making something, right? Dehydration synthesis is going to be how you synthesize or make a polymer from a bunch of monomers. You're going to do it by dehydration. What happens when you dehydrate? Or how do you dehydrate?
You lose water.
You're losing water, right? So dehydration synthesis is you're going to build a polymer. You're going to synthesize a polymer by losing water, right? And if we take a look, and this is where I'm really missing the chalkboard, right? But we'll use this illustration here instead. So here's my monomer, right? And remember that a monomer, even though the images are going to draw it as a simple little blob, it's an aggregation of atoms, right? And that are binding with each other to form a small molecule. And on the surface of that molecule, you're going to have hydrogen and OHs, right? All along the surface, filling valence seats, binding with the atoms that are in there. All molecules pretty much are going to have oxygen and hydrogen and as part of their molecular structure. Now, what's going to happen in the monomers, you're going to use some enzymes, and I'm going to break a bond here and remove the OH. And if I break that bond, that means whatever it was bound to now has an empty valency because they're no longer sharing electrons. That makes sense? Right? You've reintroduced instability. And over here, the polymer that I'm building, I'm going to do the same thing. I'm going to break off, maybe in this case, a hydrogen. Right? When I break that bond, I now leave an empty valency. And so by removing the OH and the H, this is now unstable, and this is unstable looking to react, and so they're going to react with each other, right? Attaching the monomer to the polymer, and what's left over? This H and this OH. Guess what it is? Water. So you've lost to water in order to attach this monomer onto the polymer. What? Water, we use it in so many ways, right? Does that make sense? Dehydration synthesis. You're pulling off of water in order to synthesize a polymer, in order to add instability to encourage that monomer to now bind with the existing polymer that you're growing. Hydrolysis is the opposite reaction. It's how you break a polymer into component monomers. And you do this all the time, right? This is what happens when you're eating, right? You eat a big macromolecule, polymers, and then in your digestive system, you use enzymes, right, to break that chop the monomers off of that polymer. That's digestion, right? And then you absorb those monomers, distribute them through your body using your bloodstream. When you deliver them to your cells use those monomers then to build polymers to build and run your body. So you're constantly running through dehydration, synthesis, and hydrolysis all throughout your body, all the time. right? So these are really important reactions. In hydrolysis, again, you're doing the opposite chemical reaction as dehydration synthesis. In this case, you're using hydro, it refers to water, and you're undergoing lysis. And lysis means to break or to rupture. Okay? So hydrolysis is using water to break the polymer. And you're doing the opposite reaction. I think it's illustrated on this side. In this case, you're going to cleave the bond on the polymer, right? Opening up two valence sheets, and you're going to use H and OHs from the cellular environment to bind onto the polymer and to the monomer to restabilize them, to fill their valence sheets back up. Right? So in either case, whether you're building or breaking down, you are using water. Right? Again, this is why water is one of the reasons why water is fundamental to life on Earth. Right? We use it all the time. This makes sense? Dehydration synthesis builds polymers. Hydrolysis breaks polymers down. If you can remember lysis breaks, right, then you can very readily remember that hydrolysis is the breaking down of polymers. Again, you do this in your digestive system all the time. That's what chemical digestion is hydrolysis or some form of hydrolysis. Cool? All right. So we've looked at this diagram before. We said carbon's pretty important in our bodies. It is one of the ways that we build the backbone for many of our organic molecules, right? So organic in this case means it is going to contain carbon. So I'm going to do a quick poll, right? And my poll question is going to refer a little bit back to what we learned before. If carbon has an atomic number of 6, how many bonds is carbon going to form? How many bonds is carbon going to form?
4.
So poll. Answer the poll.
See the poll on my screen?
Is it under your, sometimes if you move the tile with the images? Does it move if I move it for you guys? It does, okay.
I still don't see it on my screen, sorry.
Yeah, I don't know why. You're going to drop me an email and name the poll, that way I know to add you in.
Send you an email, you said, about the poll.
About the carbon poll. That way I know which one.
And my answer?
Mm-hmm.
Okay, I'll do that right now.
Okay. Last call, poll. Last call. Going once. Going twice. All right. So good job. You guys are getting much closer on being able to figure out those chemical bonds. Again, you've got a couple questions like this on your homework assignment. You should maybe come to office hours today at 11 if you're still struggling with how to determine how many bonds are forming. But overall, it's looking like we're getting really close on understanding that. So I can get rid of that for a second. We can go ahead and start talking about the categories of organic molecules and why carbon is at their backbone. So we're going to be talking about carbohydrates, proteins, nucleic acids like DNA and RNA, lipids, and I'm actually going to add hydrocarbons, right, which is another important carbon-based molecule. Am I doing on time? Excellent. All right, so carbon, we said, has an atomic number of 6, right? So for a review, for those of you guys that are still struggling, right, the first orbital is going to house the first 2 of those 6. The remaining 4 are going to go into the p orbital. Remember that your p orbital houses a total of 8. If you put 4 in there, right, that means you have 4 empty seats. Those empty valence seats in that outermost shell are going to want to be filled in order for carbon to gain stability. So it's going to want to form 4 chemical bonds in order to gain stability, right? And it turns out that 4 is the Goldilocks number, right? So 4 is not too many that it would be way too crowded to be able to form 4 bonds. So it's not too crowded. And 4 is also going to allow you to be able to form complex molecules, right? So it's going to allow you to form lots of branches and side chains. Right? So carbon is magic, right? And that 4 allows you to do build a diversity of molecules. And we'll talk about why diversity of shape is important in a second. So here's the deal, right? We're looking at an illustration of this is a hydrocarbon, a molecule that has only carbon and hydrogen. And the least complex of our macromolecules. And if you take a look, notice how when you illustrate the carbon, right, each carbon is forming 1, 2, 3, 4 bonds. Right? The N carbon's got 3 bonds that it's forming with hydrogen, right, and 1 bond that it's forming with a neighboring carbon, right. And then these neighboring carbons, right, are forming bonds with each other, right? And then also with a couple hydrogen, but they still have 1, 2, 3, 4 bonds. Now, what's key here is notice how it's illustrated in the staggered form. Yeah, up, down, up, down. It turns out when carbon bind to each other, they form this, what's called a pleated sheet, right? So it's almost like you folded a piece of paper and then unraveled it and it still has those little bends in it, right? Pleated sheet. So it staggers naturally when they bond to each other. And that gives the molecule 3-dimensional shape. Again, shape, we're going to learn in a second, is critical to function. Right? And then if we take a look at this illustration over here on the right, notice how you can get different shaped molecules depending on how many carbons you put, whether you put 1 carbon or 2 carbons or 3 carbons or 4 carbons. Right? And carbon molecules, you can also start to build side chains. And you can put the side chain on the 1st carbon, on the 2nd carbon, on the 3rd carbon, on the 4th carbon. Ooh, on this top side, or ooh, on this side over here. The number and diversity of molecules that with different shapes that you can form, right? It's almost infinite. Right? And sometimes you can form double bonds, and that's going to give the molecule a different shape. Right? And when you have 6 carbons in a watery solution, they're going to bind in this form to make a ring. Right? Whoa! Right? All kinds of different shapes with carbon are possible because 4 bonds happens to allow you to form all these complex interactions, right? Not too many, not too few. And what we're going to learn that's really important concept in biological systems is that at the molecular level, at the cell level, shape matters because shape is how molecules are communicating in your body, right? Shape is how your cells recognize different molecules. They don't talk, right? And if your cell needs to uptake a carbohydrate, right, it's going to be able to recognize carbohydrates based on shape, right? So the more shapes that you can form, that means the more diversity and functions you can have. And carbon-based molecules, right, are diverse in their shapes, which allow us the ability to have all the diversity of functions that we need to build and run complex bodies, right? So shape matters, right? So here's just, I'm going to show you really quickly a few different representations, right? So this is a ball and stick model of carbon-based molecules. The darker, larger shaded molecule here is the carbon, and then the smaller ones are the hydrogens, right? And so it's showing you the 1, 2, 3, 4 bonds here. Right? So just really quickly to take a look at the diversity of 3-dimensional shapes, right? To remind you that molecules are 3-dimensional in shape, right? So here's a longer chain with carbon at each branch here. Look at all the different shapes that you can form with carbon backbone, right? And these are just molecules that have only two elements, carbon and hydrogen, and they can form a diverse number of shapes, right? Then we can start to add nitrogen in there or oxygen in there or other chemical elements to add even more diversity in shape and reactivity, right? So carbon's a backbone, right? And then we can start to tweak with that, adding different chemical elements to change the properties of the molecules. Right? All right, so I've convinced you that carbon's super important, critical for life, right? This is why we say we're a carbon-based life form, right? Because our big organic molecules are fundamental to how we work. This is why we're looking for carbon and hydrogen on other planets, is to see if there's life like ours in other places. Right? All right, so here are the big 5 molecules that we're going to talk about today, starting today and into probably bleed into Thursday. For each of the organic molecules, you need to know the term for that category molecule. You're going to need to be able to describe the properties, right? And the monomer of each of the molecules categories. And then we're also going to talk about their biological significance. I'm going to give you some examples. This pattern sound familiar? Same thing you did for water, right? So as you're studying, you should be able to name off, right, the properties of water. Once you have all the categories of properties of water, their descriptions, right, their biological significance and their examples. Then you're going to run through and start studying the same thing for organic molecules. We're going to learn hydrocarbons, carbohydrates, lipids, proteins, nucleic acids. Start with the categories and then add the details. Right? Once you've got the categories down, right, it's 5. You can do them on one hand. Once you can list them off, then start adding their monomers and their properties and their biological significance, right? Study in layers. I will always recommend you study in layers. It's really important for keeping you from getting overwhelmed. And
Can I ask a question? Sorry to cut you off. So I'm assuming probably not, but do you happen to provide a study guide?
I do.
Oh, okay.
Where can you guys find the study guides? Anybody remember?
On your website?
Mm-hmm. On my Instruct website.
Okay, awesome. Thank you.
Yeah. All right. They're just guides, right? So you're going to see the same, can you identify the categories of organic molecules? What are their properties? What are the biological significance? They're not all inclusive, but they should hopefully help tailor. And the PowerPoints are on there as well, right? So we're going to start off with hydrocarbons, and these are the least complex of our organic molecules, and they are less complex because they only have 2 elements, carbon and hydrogen, right? But that doesn't mean that they don't have diverse functions, right? Because you can build a hydrocarbon with 1 carbon, with 2 carbons, with 3 carbons, with 4 carbons, with thousands of carbons, right? And you can have branches. Right? These are all linear hydrocarbons, but you can also have hydrocarbons that have branching. So you've got a lot of diversity of shape. And again, diversity of shape means you can have a diversity of function, because for each shape, you can use it for a different function. Right? I tend to avoid the term simple because it implies less good. They're great molecules. They're really important for life. They just happen to be less complex than some of the others in that they only are formed of 2 elements, carbon and hydrogen. Right. For physiological, biological significance, we're going to learn that hydrocarbons are a very simple, easy way for storing energy. Remember that when you make a chemical bond, right, you're storing energy, potential energy. And when you break that bond, exciting those electrons out of their orbitals, you release energy. Right. So hydrocarbons can be used quite frequently for energy sources, right? So if you take a look at, I don't know what happened with these images, they are now seeming to overlap. So methane, propane, ethane, butane, gasoline, diesel, what do all these hydrocarbons have in common? What do we use them for?
Fuel.
Fuel. We're breaking their bonds to get work, to accomplish work, right? Whether that's the work of heating, right, to grill your steak, to light something on fire like a campfire, butane, diesel to run a truck, right? You're using hydrocarbons for energy, right? And if we take a look, right, in gasoline and diesel, there's a lot of bonds there to break, a lot of potential energy. Right? And then, you can maybe make plastics, which are crazy hydrocarbons as well. So they can be used in energy storage or harvesting of energy. They can also be used for communication. Hydrocarbons are used in all kinds of complex ways. So we've got ancient lineages of organisms like silverfish and ants. Social insects oftentimes use hydrocarbons as a way to indicate being from the same colony or communicating different messages. And they can use hydrocarbons in complex patterns to be able to communicate. And I'm really bummed because my YouTube link is no longer working for my video on this. So, but ants can communicate between each other by the hydrocarbons that they have coating their body. So different castes or different jobs within the ant community are going to have different profiles of hydrocarbon, and this is how they recognize each other. This is also how they recognize ants that come from a different colony, an enemy, right? It'll have a different hydrocarbon scent. And this is also something that can be manipulated, right? So there's these silverfish which will rub hydrocarbons on their bodies from the ants. And that way they'll be able to invade and live inside the ant colony, take advantage of their food resources, be protected by the other ants because they're tricking the ants into thinking that they are one of them. The ants don't recognize the difference in shape. They recognize the same hydrocarbon smell that is the smell of individuals of their colony. It's kind of fun, all the different ways that organisms can interact with each other with using hydrocarbons, pardon me. All right, good. Hydrocarbons are also really important because they're a building block molecule, right? You can now pop off a hydrogen from one of those hydrocarbons and add an oxygen, and you've now changed the properties of that molecule, right? So you can use hydrocarbons to build your other types of complex, more complex organic molecules, right? So by popping off a hydrogen and adding another new cluster of atoms onto that hydrocarbon, you can change its properties, you can change its reactivity, you can change the category of molecule that it's in, right? And that's because what you're doing is you're adding a functional group, right? And a functional group is a group of atoms that are reactive, right? Or have unique properties on a much larger molecule, right? So they influence the properties of the molecule, the reactivity of the molecule. So for example, down here, I can take a basic hydrocarbon that only had two carbons. I can pop off a hydrogen and add an OH. And now this hydroxyl group converts that hydrocarbon into a carbohydrate, right? What do we know about oxygen? What did you guys learn about oxygen? So one thing I asked you to remember about oxygen.
It's important in all life.
It's important to all life. What did you learn about its properties, about its reactivity?
Oxygen's really reactive by itself.
Mm-hmm. It's really electronegative, right? So it's going to hog electrons. So I can take a hydrocarbon that's nonpolar, that's hydrophobic, and if I pop off a hydrogen and add an OH, I now have a greedy oxygen that's going to hog the electron in this chemical reaction here in this bond. I've now added polarity to a molecule that was previously nonpolar just by adding an oxygen. Right? Does that make sense? Do I change the reactivity of this molecule now? Yes. And I can change it from being hydrophobic to being hydrophilic just by adding an oxygen. Right? So swapping out a functional group has a big influence on a molecule. And different categories of molecules are going to have different functional groups. So for example, over here, I can take a 1 carbon hydrocarbon, pop off a hydrogen, and add this N nitrogen with 2 H's, what's called an amino group, right? And now I just made amino acid out of a hydrocarbon, right? By changing the functional group, and I can use it to build a different category of molecule, a protein in this case, right? So functional groups are a really important way to change the properties and reactivities of a molecule. It's also going to tend to be the reactive part of the molecule. Good with functional groups? Any questions, functional groups? All right. So hydrocarbons we've covered, check, right? First organic molecule, we've got 4 to go, right? Next category is a carbohydrate. And this one's usually pretty straightforward for you guys because you are already familiar with the term carbohydrate. It's not a new term for most of you guys, right? So we are going to talk a little bit more about the complexities of carbohydrates. So carbohydrates are sugars, right? Their monomer is going to be what we call a monosaccharide or a simple sugar, one sugar ring, right? That's the monomer, is a monosaccharide. Mono means one, saccharide means sugar, one ring of sugar. So carbohydrates, those sugars, right, are going to be a 6 carbon molecule. And remember that in aqueous solutions, in watery solutions, like inside of our cells, outside of our cells, those 6 carbon molecules are going to form a ring, right? So those monosaccharides are going to be in this ring shape, right? And you're always going to see them illustrated like that in terms of in their biological context, right? So it's that monosaccharide is going to have some oxygen on it. You don't need to memorize the molecular structure or formula for monosaccharides, but I do want you to know that it has oxygen on there. So what does that tell you about the properties of those monosaccharides?
Unstable.
Well, they can be reactive, right? They're going to have polarity associated with them. What do you guys think? Hydrophobic or hydrophilic carbohydrates?
Hydrophilic.
Hydrophilic. They're going to dissolve well. You know this already, right? Because when you put sugar in your coffee or your tea, it dissolves readily, right? It's hydrophilic. Good so far? And you already know that sugars are energy sources. Look at all the chemical bonds in there that you can then break apart, right, to release energy. And in fact, we're going to talk about carbohydrates and sugars and how important they are in cycling cellular energy. You're going to use them for making or cycling ATP. But that's a next unit too, right? So a carbohydrate can be one simple monosaccharide. If I put 2 together, ooh, how would I attach 2 monosaccharides together? What's the chemical reaction that I would use? Oh, that would have been a good poll question. This morning before lecture, I'm like racking my brain for good poll questions, and they seem to pop up easier in the what do you guys think?
Dehydration reaction.
Yeah, dehydration synthesis. It's going to be used to put one monosaccharide and bind it with another. And that's going to make a disaccharide, 2 sugars together. And then if I keep adding anything more than 2, I can refer to as a polysaccharide, 2 or more sugars in a chain, right? So it could be 3 or it could be 300,000, depending on the molecule. They're all polysaccharides. Make sense? Right. All right, so let's take a look. The monosaccharide, right? It turns out that, so if we take a look at the structure, remember that at the joint, there's a carbon, right? At each little node here. represents the carbons, right? And then here you can see all the little functional groups at each corner of the corner of the molecule, right? Are attached to each of the carbons. Now, if I put, say, let's go with just this single oxygen, right? I can put that single oxygen on this corner here and make a fructose, right? And then if I move in the position of the functional groups, maybe now instead of at this corner, I put the oxygen over here. That's a different shaped molecule, right? That's a different molecule. That's a different type of monosaccharide, right? Or if I put the oxygen on this corner, or this corner, or this corner, they're all different versions of that monosaccharide. So my point here is, depending on the order of the functional groups, you can make different shapes of the molecule depending on how you assemble all the same types of atoms, but in different combinations, right? To make different monosaccharides. So here we're taking a look at fructose, which is a monosaccharide. So is glucose, so is galactose, right? But how the functional groups are organized are different, giving the molecules a different shape, therefore different properties. Right? And indeed, different monosaccharides are going to interact differently with, say, the taste receptors on your tongue, right? And have different, you're going to detect, therefore, different sweetness, right? So some are going to be more sweet to you, some are going to be less sweet to you, and your tongue is recognizing the difference in molecular shape. Does this make sense? There's different types of monosaccharides. You don't need to memorize, right, their molecular structure and how their functional groups sit. But you should know that glucose is a monosaccharide. You should know that fructose is a monosaccharide. Right? There's a couple examples that we'll be using throughout the semester, right? So glucose, we're going to talk a lot about. That's the cellular energy source. That's the sugar you're going to be using for cycling ATP. Fructose, anybody know where fructose comes from? It's a type of sugar that you see frequently in fruits, right? Is fructose. When you're eating a sweet peach, you're detecting the fructose in there. And notice how they all the monosaccharides are going to have 6 carbon, 12 hydrogen, and 6 oxygen. They all have the same atoms, the same numbers, but their organization is different, making them different, right? So we call this isomers, right? So here you can see the difference. If we straighten out that molecule, you can see that the difference between glucose and fructose is where we put that double bond with the oxygen. So by changing the position of that functional group, we've now changed the shape of the molecule. We can use fructose for a different function than we use glucose.
I have a question. So
Yeah.
from this picture between this glucose and the fructose, so like just from the what it looked like, can we like get some conclusion about the two things, like difference? Like, for example, is fructose is more stable than glucose, or?
They're equally stable, they're just different shape.
Okay, got it.
And if it's a different shape, you can use it for a different function.
Got it, thank you.
And you're going to be consuming both, right? So when you think about high fructose corn syrup, right, you've modified the proportion of glucose and fructose that's in that, usually cornstarch. Right? So fructose has a higher sweet index. It tastes sweeter to your tongue, right? Which is why, one of the reasons why it's not the best thing for you. But that's another lecture. We'll maybe try and get to that in nutrition. Okay, so again, dehydration synthesis, we put 2 monosaccharides together and we get a disaccharide, right? Now, disaccharides can be different depending on which 2 monosaccharides you use, right? So here's sucrose, it's a glucose and a fructose, versus lactose, right, which is a galactose and a glucose together, versus maltose, which is a glucose and a glucose. Right? They're all disaccharides, 2 sugars, right? But you're going to have different types of disaccharides, right, depending on which 2 monosaccharides you use to build them. Make sense? Simple sugars, right? So sucrose we're used to using, right? We use sucrose usually as table sugar, right? So you're familiar with sucrose, even if you didn't know its name, right? Anybody know where we get lactose? Where do you find lactose? Does that sound familiar?
Milk.
That's the sugar that's in milk, right? So milk tastes a little bit sweet to you, as it turns out, right? And then maltose. Does that sound familiar? That's the sugar that's in grains. And so when you ferment those grains to make malt beverages, right, like beer, right, maltose is the sugar, right? That's found in beverages that have come from grain products, right? So it's a sugar. You can break it apart and extract the energy. Hey, no, come here. Somebody woke up. So sugar sources, energy sources, you can break them down and use them to cycle energy, right? And this is why beer gives you beer gut, right? It's got a lot of sugar associated with it, right? Beer bellies, right? All right. Again, a polysaccharide, 2 or more, right? So monosaccharides and disaccharides are usually simple carbohydrates or simple sugars, right? They break apart very easily, right? Once you consume them, they break apart and you can absorb them and process them very quickly. A polysaccharide, right, is 2 or more than 2 monosaccharides in a chain, right? These are also great energy sources. They have, in fact, more energy because they have more sugars along that chain, more chemical bonds to break, right? So examples of polysaccharides that we're going to be learning are starch, glycogen, and cellulose, right? Starch, ooh, does that sound familiar, that term, starch? Where do you get starch? Nobody? No starch?
Cornstarch.
Cornstarch, yes.
Bread.
Bread. What else is starch? Potatoes, yeah. So starch, right, is a molecular savings account. Right? Sometimes polysaccharides can be used as a way of storing energy. Right? In plants, they're going to store energy, sugar energy, so they're going to photosynthesize, make a bunch of sugars, and then store them in long chains called starch. That's their plant savings account. So like in a potato, that's a root structure where that plant is storing sugar energy to overwinter. And then in the spring, when it regrows, it's going to break that starch down into component monomers using which chemical reaction? Hydrolysis, to use that sugar energy to regrow the next spring. Right? So starch is a energy storage for plants. Right? We consume those plants or that starch as an energy reserve in your digestive system. You're going to be using enzymes to hydrolysis that starch and release those sugars that you can absorb and deliver to your cells. Does that make sense? Storage of energy, lots of energy in starch. Right? Now, if you as an animal eat starch, right, break it down in your GI tract, it's going to absorb into your blood vessels, get delivered to your cells. Any extra sugar from that broken down starch, you're going to store in your molecular savings account a polysaccharide called glycogen. So animals store sugar in a molecule called glycogen. Right? You build glycogen by dehydration synthesis, and generally for animals, we store it in our livers. And then when you need it, you can hydrolysis, break it apart, and release it back into your blood vessels, right? Like when you're exercising. One of the things that happens is glycogen in your liver gets hydrolysis, chop, 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 and released into your bloodstream so that you have the sugar to cycle ATP to allow you to fuel your exercise. Right? So again, we're constantly building and breaking down polymers. Good? Make sense? Yeah. Storage molecules. You can also use complex carbohydrates, right? Like for structure, right? So cellulose is what we're going to talk about next. And plant use cellulose for structure. And I've got just a corner of a cellulose molecule here. It's a really complex carbohydrate. It is not only one chain of sugar rings of monosaccharides, it's 1, 2, 3 chains, then bound to each other, right? And these bonds that bind one chain to another are super unique in the molecular world. There's only one organism on Earth that can break down those bonds between cellulose and liberate the sugars to be used as energy. Bacteria. Well, maybe a couple fungi can break down cellulose as well, right? In our digestive system, it just moves through. We can't break the cellulose bonds down and release the sugar, right? This is fiber in our diet. Plants use cellulose for structure. So around every single cell in a plant body, you've got cellulose. Right? And again, it's got oxygen all over it, so you know it is hydrophilic. Right? So plant walls, right? Plant tissue, like we break it down and make it into pulp to make paper towels. Right? That's cellulose. And it's hydrophilic, bounty, quicker, picker, upper, right? Because of the oxygen that's in there. It's the polar nature of the cellulose molecule. Cotton towels, we already talked about. Make sense? Paper, right? Like your notebook. Good? Cellulose, complex polymer for structure. Any questions? No? No questions? All right. So cellulose is just plant cell walls. I have a funny story. Do I have time? So, I'm constantly telling you why learning biology is important so that you don't spend wasteful money buying marketing ploys. I used to have this neighbor, and she was still actually a close friend, but she was a belly dancer, right? And she had this other woman in her belly dancing troupe who was a total stoner, right? And this gal comes over and she's like, "Oh yeah, I smoke a lot of pot, but I do it healthy." I'm like, "What are you talking about?" Right? And she's like, "Oh yeah, I get these special papers to wrap my joints. They're made out of cellulose." And so she's paying like $25 for this pack of joint wrappers because they're cellulose. And I was like, "Are you kidding me? Do you know what cellulose is? That's just paper that's been processed in a different way to get you to pay $25 instead of, I don't know how much does a pack of papers cost, $5. So cellulose paper, right? Cell walls. I thought it was kind of funny. My friends are so sick of me. Constantly like, I don't have time today, but remind me later and I'll tell you the story. I had a crazy book club conversation about hypnosis the other day, and I was just like, just hold your tongue, Letitia, don't even bother. All right, so lipids, right, are our next category. So far we've talked about hydrocarbons, we talked about carbohydrates. Carbohydrates we're going to come back and learn a lot more about. Next category, molecules of lipids. Now the lipid category is the oddball category, because it is the only type of organic molecule or biological molecule that does not have a characteristic monomer, right? So structurally, it's a little bit different, right? So lipids are going to be categorized based on their properties instead of their monomer type, because again, they don't have one characteristic monomer. They're a little bit more diverse in their molecular shape. What's going to combine them all together, group them into this category of lipid, is how they interact with water. They are all hydrophobic, right? Water fearing, which tells us that they are non-polar for the most part. Yeah, does that make sense? Nonpolar. So this is going to be things like fats, oils, steroids, phospholipids are all categories of lipids, right? And if we take a look at the fat structure here, right? So we're going to have this glycerol backbone. You can see there's the carbon, right? And then it's got these fatty acid tails, right? And I'm going to talk about this a bit because we're going to spend some time learning about what the heck is a phospholipid. It's an important category of molecule that we're going to be talking about next unit as well. So these fatty acid tails and lipids, right? Each little joint here, each corner represents a carbon, right? And remember, it's got all kinds of other molecules attached to it, usually hydrogen, right? So there's the little carbon backbone that you can see all the length here. Now, in this big fatty acid tail, you've got a lot of chemical bonds, which means you've got a lot of potential energy. So lipids are very energy-rich molecules because they've got a lot of bonds that you can break to harvest that energy from. All right. So lipids diverse in their function, right? So you can have things like lipids like adipose tissue that's used for insulation, right? So here you're looking at these seals, right? And they're all chock full of blubber, right? Which is adipose or fatty tissue. And the idea there is that, you know, it's an energy reserve that they can use when times are lean, but it's also really important for insulating them, allowing them to survive in that cold ocean, right? So it provides a fatty layer that minimizes heat loss, right? Lipids can also be great dietary energy sources, right? So they've got 2 times more energy than carbohydrates or proteins because of all the bonds that are in those fatty acid tails. The 2 types of dietary lipids that we generally consume are fats and oils. I'm going to talk a little tiny bit about their molecular structure and what makes them different. Right? So fats, we're going to learn, generally come from animal sources. Right? It's a type of lipid that's used in animal bodies to store energy. It's generally solid at room temperature. It's generally solid at room temperature. Oils, on the other hand, are generally liquid at room temperature, and they generally come from plant sources, right? So an example of a fat would be like lard, right? Butter, right? Animal sources. Oils would be like olive oil, canola oil, sunflower seed oil. They're generally liquid at room temperature. Can you think of any exceptions to that rule? Coconut oil, right? So I'm going to talk about that a little bit more later on. You can change the molecular structure of a plant oil and make it solid by a process known as hydrogenation, right? And I'll talk about that a little bit later on. So lipids are one of those molecules that are current day highly vilified, right? There's all kinds of fat-free diets, right? They are a great energy source, right? And your body rewards you with dopamine in your brain, a pleasure hormone, for eating them, because lipids are not super common in the environment, right? When you think about foods that you can go out to the garden and pick, very few of them, right, have a high lipid content, right? Maybe avocados, coconuts, and nuts, right, might have high oil content, high fat content, but your cucumbers, your tomatoes, right, things like that don't have a high fat lipid content. So when humans were evolving on the savannah, say, when you came across a high lipid food, oh, your brain rewards you with lots of dopamine, right? Nowadays, right, we consume way too much dietary. Some stranger danger out there. Nowadays, when we get lipids, right, your brain's still giving you the eat more, eat more, eat more, and we end up consuming way more than we need, right? So this is where lipids get their bad rap. Does that make sense? A little bit of lipid, though, is important in your diet. You need those fatty acids for building cellular structures. Lipids, fat in your diet flavors your food, right? It also is really important for that satiation process, right? So when you're consuming a meal, your GI tract is detecting what's in that meal. And when you've gotten some lipid in there, it triggers to your brain telling you, okay, we've got enough calories, feel full, satiation, right? So when we do those fat-free diets and don't have any fat in our meals, our brain is registering, oh, there wasn't enough calories in that meal. Eat more, don't feel full, don't feel satiated, right? So a little bit of lipid in each meal is very important, right? Because it also, in addition to flavoring your food, it triggers satiation, and it also helps to regulate the rate at which food is moving through your digestive system, so allowing you the benefit of extracting more nutrients out of that meal. Make sense? Yeah, lipids. Okay. So how many of you guys heard of good fats versus bad fats? We hear that a lot. So let's talk about the types of fats, what makes them good versus bad, right? Again, vilification of molecules. So we're going to talk about what are called saturated fats and unsaturated fats, right? Saturated fats are animal fats generally, right? Lard, butter, solid at room temperature. And when we talk about saturated fats, the term saturated is referring to their molecular structure. Right. So here's the backbone of the molecule, and here's the fatty acid tail. Right. Whoa, look at all those bonds in there. Lots of energy. And notice that for those carbons, every potential spot that they can have a hydrogen, they do. No double bonds, in other words. Right. They are saturated with hydrogen. This is where the term comes from. And when they're saturated with hydrogen, they form that pleated sheet, but overall, the fatty acid is pretty flat. Right? It's pretty flat. Right? And I'll talk about why that's significant in a minute. Versus unsaturated fats. Right? And unsaturated fats, we usually call them monounsaturated, diunsaturated, polyunsaturated fats. What the unsaturated means is that there's some hydrogen that have been popped off, and carbon are forming double bonds instead of bonding with hydrogen at every possible location. So they're not saturated with hydrogen. They've got some double bonds. Monounsaturated means one double bond. Diunsaturated, 2 double bonds. Polyunsaturated fats, 3 or more double bonds. Right? And notice what the double bond does, right? A double covalent bond literally pulls the atoms closer together, right? And creates a little tweak or bend in the molecule. We call them kinky, right? Makes this little kink here, right? Unsaturated fats, right? Olive oils, nuts, nut oils, avocado oil. These are the types of fats that are in plant sources generally, right? Liquid at room temperature, right? Because they don't congeal as readily. Make sense? Has to do with their molecular structure. All right, so saturated fats, here's the idea, and the science behind this, right? But it seems to be one of those things that kind of stuck for folks. The idea is that a high saturated fat diet, right? Turns out any high fat diet is going to do this, right? Those lipids are going to, those hydrophobic lipids in your hydrophilic bloodstream are going to be repelled, right? And they're going to build up, stored in the walls of your blood vessels, right? So fats can get stored in the walls of your blood vessels. Arteriosclerosis, for example, is one of the ways that fats accumulate along with other minerals in the walls of your blood vessels. And as they accumulate in there, right, it narrows the diameter of your blood vessel, right? So here's a normal blood vessel, and here's a vessel with arteriosclerosis, all this like lipid and mineral built up in the wall of vessel, reduces the flow of blood, puts a strain on your heart, right? Because your heart has to beat more frequently in order to pump equal volumes of blood. I'll talk more about the health consequences of this later on. Right? So the idea is that the saturated fats, because they are flatter, right, they stack up more readily in your blood vessels, right? Versus the unsaturated, which have that kink in them, they don't stack up and accumulate as readily. They slough off from each other. Right? I always use the example of, like, if I took a flat folder, right, this would be like a saturated fat. I could stack them to the ceiling. Right? Whereas a unsaturated fat would be like something like a binder. I can't do it. I can't. Would be like a binder, right? It's kind of kinky. Can you stack binders to the ceiling? Less stable. They tend to slide off each other. So that was the idea behind saturated fats are bad for you. Unsaturated fats are better for you, right? It's a gross oversimplification of how those fats interact with your body, right? But we now know that saturated fats are fine for you as long as they're in moderation, right? Make sense? What's the exception to the rule? Can you guys think of any vegetable or plant fat source that is solid at room temperature?
Avocados?
Potato?
Oil, plant oil that's solid at room temperature.
Coconut oil,
Coconut.
you just mentioned?
Yeah, coconut oil. Yeah, coconut oil. All right, so you know how there's, I'm going to leave you with this. You know how there's that big coconut oil fad? Oh, it's so healthy for you. It's healthy. Oh, use coconut oil instead. Lather it on your toast. Lather it on everything that you're eating. Right? So remember what we said, saturated fats, animal sources, flat, right? Stack well, not good for you. Not good for you, right? Unsaturated fats from vegetable sources have that kink. Healthier for you, right? Eat more vegetable oils. Choose them over animal fats that are saturated. So this is where, use coconut oil because it's a plant source, therefore it's healthier for you. Except that, what do you think makes it solid at room temperature like animal fats? Its molecular structure is saturated. So it's a plant source that's saturated fat, making it pretty much the same as butter. Right? So the extrapolation there was, vegetable oils, vegetable plant sources of fats are good for you, right? Because they're unsaturated, but they left out the step where, except for coconut oil, which is saturated. So this is where the, understanding biology a little bit is beneficial, right? Because now you have all these folks over liberally using coconut oil because they think it's healthier, right? When in fact, it's just the equivalent of any other saturated fat pretty much, right? You might as well be slathering that much butter, right? Make sense? Oh, biology, chemistry. Right. All right, I think I'm out of time, yeah? I just don't have time for waxes. Okay, so I'll finish up the rest of, do I have 5 minutes left or I am out of time? You know what, I'm just gonna stop here. And I will finish up the rest of lipids when we come back on Thursday. Yeah, I think I'm out of time.